Hey guys, so I wanted to talk to you a little bit about the cell cycle and show you in a drawing what is it all about and what's really important to remember about it. So the first thing is to realize that the cell cycle, although it's a cycle, so there will be a big circle, right? There is a division, a major division that occurs. And the first one is the division between interphase and the mitosis. So the interphase will take most of a cycle. The interphase is between two different phases. So interphase in this case is between two different mitoses. And so your interface is then subdivided in three different sections. Four if you include G0, but we're just going to focus on the first three. 1, 2, and 3. So that would be G1, that would be the S phase, and that would be the G2 phase, right? And of course, the G0 phase, which we'll put down here for now to be able to see it on the board, it's really a phase that cells go in and out. Just to make sure we finish the cycle, all of that here, this is the mitosis, and we'll talk about it in another video. Okay, so now that we know all that, just I want to point out really important things. Most of the time, cells spend time in the interface. So you would have a hard time finding cells in mitosis. It's rare, but it exists, and of course it exists. But it's really most of the time in interface. And even more importantly, it's most of the time in G0 phase. The G0 is when cells are just going through their function. They just don't duplicate the DNA, they don't get ready for mitosis. That's what all the cells in your bodies are mostly into. That's the G0 phase. When they switch to G1, what they do is they prep for the S phase. So keep in mind the S phase, it stands for synthesis. So S for synthesis, and let's write it right there. It's synthesis of DNA. And during that phase, the DNA is doubled. That's when DNA replication takes place. That's what we've seen last week with DNA replication. So the G1 is really all prepping for that replication. So it's making proteins. Which proteins, you ask? Well, the DNA polymerase, the binding proteins that hold the DNA, the ligase, all of those proteins we talked about last week. That's when G1 produces them. Then when you go to G2, you now are after replication, you now are replicated chromosomes. So keep in mind, G1, G0 chromosomes are single strand. So one the homologous chromosomes will be like that, right? Whereas in G2, they are replicated. So there will be little H's, right? Because you did DNA synthesis. And then when you start from mitosis, there's a whole system that's going to separate the sister chromatids. And we'll talk about it in another video. But really, this is the gist of it. Make sure you remember that most of the cells are in G0 all the time. When they go into G1, they prepare the molecular components necessary for synthesis. That's DNA replication right there. So you go from a diploid cell with one copy of each chromosome to a diploid cell with two copies. So you just double the number of molecules of DNA. Well, that's what it is here in G2. That's the replication we saw last week. And all of those four phases, G0, G1, S, and G2, it's all interphase. And then there's a mitosis here. Okay, that was all.